Hi everyone, and welcome to another SYCL 360 edition. It's my pleasure today to be here with Dr. Alan Remaley, Senior Investigator of the National Heart, Blood and Lung Institute of the National Institutes of Health. Welcome, Alan, and thank you for joining me today.
Great, yeah, thanks for the invitation.
Alan, would you mind start describing to us what you do at the National Institutes of Health?
Great. So I have basically two positions. I'm a senior investigator, as you said, at the NHLBI, where I do both basic and translational research related to lipoprotein metabolism. And as you know, I'm trained as a pathologist. I'm also a senior staff member of the Department of Laboratory Medicine, where I oversee part of the clinical laboratory related to clinical chemistry, and particularly interested in cardiovascular biomarkers, which I'll be talking about later today.
And how did you come across your line of work and research interests?
So I grew up in Pittsburgh, Pennsylvania, where I went to medical school, and I still remember vividly. I was, I think, a third-year medical student, and someone asked me about the clinical findings for Tangier disease. And I hate to say, at that time, I knew very little about Tangier disease. And the attendant came up to me and says, "Alan, don't worry, no one knows anything about Tangier disease." So I got intrigued, and I read about Tangier disease, which is a rare disorder of HDL metabolism. It was discovered on Tangier Island in the Chesapeake Bay, and was discovered by my predecessors at NH, Don Fredrickson and others. So from that point on, I got interested in lipids from just a random question that I didn't know the answer to at the time. And then in 1998, our lab and many other labs, unraveled the cause, and I've worked on the ABC transporters and HDL metabolism and other aspects of lipoprotein metabolism since then.
Wow, and we know that that's been a changing field in laboratory medicine. There's been a lot of new recommendations in general for lipid testing on patients, and so I'd like to get your input on what are some practical recommendations that we can give today to young lab directors who are interested in revisiting perhaps what they're offering in their lipid panels.
Well, I think the most important thing is to follow the guidelines, and so they're called the Multi-Society Guidelines, led by the American Heart Association, National Lipid Association in 2018. And you need to be able to offer a lipid panel that's reliable. And the big question there is what's the best way to measure LDL, whether you should calculate it by the Friedewald or the Martin equation. We are working on some new ways to do that. But measuring LDL or estimating it accurately is important because we have new agents now that are very effective in lowering LDL. So that's important. So I think following the guidelines and also probably offering ApoB, LPLA, CRP. So the new guidelines suggest that there's a large number of people who are at intermediate risk, and there's a dilemma about what to do with them. One could actually get a calcium score and has a very high negative predictive value, but it's also very valuable to look at these other so-called risk enhancer tests. So I think the best thing a laboratory can do is actually to offer the tests that are recommended in guidelines and make sure that their assays work as well as they can. I would be a little bit cautious, although some of the, I think, direct assays are good, but the direct assays are very popular, both direct HDLs and LDL. They've been around for at least 10, 15 years. There were some problems, they're better, but they don't all work, they work reasonably well, but on some rare diseases, like I study LCAT deficiency, one of the particles accumulates is lipoprotein X, and that can give you a false signal. So one has to be aware of the limitations of whatever assay you offer.
Excellent. Well, thank you for that input.
Right.
And I'm curious, then you mentioned that you're currently working on a new LDL equation. Can you elaborate a little bit on that and other things you're currently doing?
Sure, yeah. So the equation that actually most people still use to this day was developed in 1972 by Friedewald is the first author, but Don Fredrickson is a senior author. And it's a relatively simple equation, and it's really stood the test of time. There's been at least a dozen equations, but the Friedewald is still probably one of the more robust equations. But there's a limitation largely related to the estimation of the LDL cholesterol relates to high TG. So a major breakthrough was maybe about 5 years ago. Seth Martin and his colleagues at Johns Hopkins came up with a table, essentially, that adjusts the factor to divide by based on not only the triglyceride level but also the non-HDL. And other labs are starting to implement that, and that's, as I said earlier, important because we know that TGs cause problems, particularly when LDL is low, because the absolute true value of LDL is quite low. So if you have a bias, which may be relatively small, if you have high LDL, when you have low LDL, it could represent a significant fraction of what you're measuring. And it can scare people away from aggressively treating patients because you can get sometimes even negative LDL values. So it's not published yet, but hopefully soon. We've been working on a new equation that's an equation. And what we discovered is by using more complicated mathematical terms, although for the end user it doesn't make a difference, we could make even a greater improvement. And what we discovered is that currently, even with this, the Martin table, you're not supposed to use the equation with TGs above 400. So we can go to TGs about 800 mg per deciliter and get this equivalent accuracy. And that represents about the 99th percentile of the population in terms of TG. So, and we validated with our partners, Jim Ontvos at LabCorp and Jeff Musin at Mayo Medical Labs, and we tested over 250,000 patients, and it works very well. Compared it also against the gold standard, a beta quant, as well as other direct assays. So it seems to be a pretty robust assay. So that's something I'm currently working on.
Excellent. I look forward to seeing that because definitely we can all benefit from the significant improvement over high levels of triglycerides. And lastly, Alan, we're curious about what's next. What are we're looking at working in the near future?
So as I mentioned earlier, I sort of have two hats, and so I'm very excited about this LDL equation and in the laboratory, but I also do more basic science. And I remember when I started my career in the early 90s, there was some skepticism about the importance of triglycerides in cardiovascular disease, or at least the lipoprotein particles. And the Europeans were always kind of pro SYCL and that the triglycerides are those particles. And the genetic studies in the last 5 years have clearly revealed that the genes that regulate triglycerides, and there's about 40 genes, but there's a handful that have profound effects like lipoprotein lipase, ApoC3, that if you have polymorphisms or loss of function, gain of function mutations that modulate triglycerides, that degree of modulation correlates very well with risk. And so if you have elevated triglycerides, you have increased cardiovascular disease. So statins are wonderful drugs, although they have problems like any drugs. But on balance, I think statins are a big advance. But what I think a lot of people don't realize is that under the best of circumstances, they only reduce events by about 30%. And I think we in the future may do better with PCSK9 inhibitors, lowering LDL even more. But there's probably some unmet medical need related to the other particles. And for one, for sure, is Lp(a). But these triglyceride-rich particles that are produced as a consequence of lipolysis are probably very pro-atherogenic. And so there's great interest now in developing new drugs. And so we've developed a small synthetic peptide mimetic of ApoC2, which is an activator of lipoprotein lipase. It also antagonizes ApoC3. So, and we have only done preclinical animal models, but we've licensed it to a company called Kidiot, and we're very interested in trying to, well, I'm very interested, whatever time I have left in my career, to try to get something that would impact in terms of patient care. So that's at this time, my favorite project.
And we hope it will be successful. Finally, I'd like to ask if you have any career advice you'd like to give to young laboratory professionals.
I think maybe two. So one is, I think for your own personal satisfaction, as well as helping people you work with, I think it's important, particularly as you get older in your career, to kind of step back and ask what is not only for me, but what's also in it for the people you work with. You know, we're each on a mission in terms of life and what we learn and what we're capable of and our potential. And I think understanding and working very collaboratively with other people is, in the end, is more satisfying to you as a person, but also the people you work with. And I think that that's something that it's easier to do as you get older, but I think it's very important that you kind of shift the focus away from yourself in terms of the bigger team. And I think the other advice, again, I think it's easier as you get older, is that when you're starting your career, there's so much to learn. And you're just struggling to learn it. But I think as you get older, you realize much of what we learned is not true. And you have to go back and think more deeply about things. And I think when you go back and revisit things, it's only really then that you truly understand it. And then many times when you do that is when you actually have a conceptual breakthrough. So I think you have to also question always. Not only question what we don't know, but question what we think we already know, because what we know sometimes is not true or is not the full story, and you can make it better.
Well, thank you for the helpful advice. And again, thank you for joining us
Right.
today, Alan. Appreciate it. And thank you, everybody, for watching.